Hi everyone, welcome back. In this video, we're going to talk about a very important organelle inside of the cell. It's got some key roles in biosynthesis and degradation for the cell and some high-yield clinical disorders. So let's talk about the peroxisome. All right, so here's the outline for this video. I'm going to start with an overview of the organelles and see where the peroxisome fits in. Then we're going to talk about the functions of the peroxisomes and its mechanisms. And then finally, we're going to wrap up with the clinical correlates, which are the peroxisome disorders. And there's 3 of them here that we're going to go through. All right, so let's start with an overview of the cell to see where the peroxisome fits in. So this is a screenshot from the video on the human cell. And in this column, we have all the important organelles and structures of the cell. In this column, each of their function. And here I have them kind of categorized into their general role for the cell. And I've grayed out everything here so we can focus on the blue here, the lysosome, peroxisome, and proteasome. These are the zomes of the cell. Their main job is to help break things down inside of the cell. So essentially keep the cell tidy and free of debris. And each of these 3 structures degrades different things for the cell and has different mechanisms. The one that we're going to focus on in this video is going to be the peroxisome. All right, so now let's talk about what do peroxisomes do inside of the cell. And unlike the lysosome and proteasome that are pretty much just for degradation, peroxisomes not only break stuff, so they're not only responsible for degradation, but they also make stuff. They also do biosynthesis. So let's talk about the biosynthesis first. And here, peroxisomes are where 2 important things are made. So number 1, bile acids. These are specifically in the liver cells. Bile acids are important because they help absorb dietary fat. These are made in the peroxisome of liver cells, secreted as part of bile into the small intestines, help absorb dietary fat. The second important thing that's made inside of peroxisomes are these plasmalogens, and these come up a lot on test questions. So plasmalogens are special phospholipids that are found in cell membranes of cardiac cells and neurocells, but they're also very abundant inside of myelin, which, as you know, insulates neurons. And this is one of the reasons why someone who has a peroxisome disorder, they're bound to have some neurosymptoms because of the lack of plasmalogens and the deficiency or abnormality in myelin as a result. So now let's talk about the breaking down stuff function of the peroxisome. And the most important thing, the main thing that they're known for breaking down is these special fatty acids, which are 2 types of them, the very long chain and the branch chain fatty acids. So the peroxisome is pretty much the only place in the cell where these 2 things are broken down. So this is their main degradation function. They also break down a little bit of ethanol and some amino acids. So how do peroxisome break things down? By oxidation reactions. And you can tell that the mechanism is oxidation because of the name peroxisome. And these oxidation reactions are going to generate a lot of hydrogen peroxide. Again, peroxide for peroxisomes. That's how they actually got their name, because there's a ton of hydrogen peroxide inside of peroxisomes. That's where they're found in large amounts. And it's not a good idea to have a lot of this around because it can be toxic to the cell. For this reason, peroxisomes also carry a lot of catalase inside of them, which then helps break down this hydrogen peroxide to water and oxygen, all to facilitate their main mechanism of degradation, which is oxidative reactions. All right, so now this process of this special fatty acid degradation through oxidation actually is pretty important for the peroxisome, so we have to talk about it a little more, which means we have to talk about biochemistry, which I know, take a couple of deep breaths, take some Zofran, but we're going to get to it together, and I'm going to make it make sense. So let's go through it together. So first, we have to do a little bit of terminology here. So fatty acids are actually named based on the length of their carbon chain, right? So if you have short ones, they're called short chain fatty acids. If you have medium ones, they're medium chain fatty acids. And you guessed it, the long ones are known as long chain fatty acids. Now, if you have very long chain, so more than 20 carbons in the chain, these are known as very long chain fatty acids. And then if there's actually 1 or more branches on the carbon chain, these are known as branch chain fatty acids. So that's what these 2 terms mean right here. Very long chain and branch chain fatty acids. And these are the 2 that we're concerned with because these are the 2 that are degraded in the peroxisome. So now, why do we need to break down fatty acids anyway? Well, it's a very important part of the cell's metabolism because by breaking down these fatty acids, it normally happens in the mitochondria through a process known as beta oxidation. The fatty acids gets degraded all the way down to acetyl-CoA. So number 1, this is going to generate a lot of energy for the cell, which it's going to use. And number 2, it's going to help prevent the accumulation of these fatty acids because if they don't get broken down, they're just going to pile up inside the cell and cause problems. Now, this process of fatty acid degradation or oxidation is super easy, very straightforward for the short, medium, and long chain fatty acids because they can just get right directly into the mitochondria, either just directly or via a shuttle. And once they're inside of here, they can undergo beta oxidation, so no problem. But this is a problem for the very long chain and the branch chain fatty acids because they can't get into the mitochondria. And so this is where the peroxisome comes in. So what ends up happening is that the peroxisome actually has special transporters for very long chain fatty acids, special transporters for branch chain fatty acids, so it can actually take them up from the cytoplasm inside of it. And once they're inside, those oxidative enzymes are going to perform oxidative reactions to the very long chain and the branch chain fatty acids and break them down. One important little factoid you need to memorize is that the very long chain fatty acids undergo beta oxidation and the branch chain fatty acids undergo alpha oxidation. So there are different processes for these two, and you need to know that. What's going to happen though is that once they're oxidized here, they're essentially broken down into short, medium, and long chain fatty acids and released back into the cytoplasm. And now here, they're just going to be taken up because short, medium, and long chain fatty acids can be taken up into the mitochondria, and they're going to undergo beta oxidation just like regular. And this is how the peroxisome helps deal with breakdown and degradation of very long chain and branch chain fatty acids for the cell. All right, so now let's talk about why all of this matters, which are the clinical correlates related to the peroxisome. There are three important diseases I want to talk about. First one is Zellweger syndrome, and this is probably the most well-known and the highest yield one. So let's start with this one. So it's autosomal recessive, onset during infancy, like most of these conditions. The defect here is going to be in a mutation of the gene, which is the PEX gene. This PEX gene normally encodes for peroxin proteins, so essentially you're going to have a deficiency of the peroxin proteins. The main function of these proteins is to literally form the peroxisome. So when you have impairment of this gene, you're going to have absence of these proteins, and therefore you're going to have impaired peroxisomal biogenesis. So it's not going to form, and it's going to be missing all of its enzymes. So this is a major mutation because essentially you have no functioning peroxisome. Therefore, some of the important labs here are going to be elevated levels of very long chain and branch chain fatty acids. Let's reason through that. The job of the peroxisome is to break these down. If you don't have functioning peroxisome, they're going to accumulate, so serum levels are going to go up. Makes sense. The other important lab value is that the plasminogen levels are going to be low. So let's reason through that. Job of the peroxisome is to synthesize plasminogen. You don't have functioning peroxisome, so plasminogen levels are going to go down. Once again, makes sense. Symptoms are going to be primarily neuro, as in seizures and hypotonia, and liver, as in hepatomegaly and jaundice. Unfortunately, this is a very poor prognosis. Treatment is supportive only. This usually causes death very early on in childhood. All right, so disease two is Refsum disease, and this one is also autosomal recessive, but this one is hugely different from Zellweger syndrome because here the defect is very specific in the peroxisome. Specifically, you have a problem with alpha oxidation. So if you remember a couple of slides back, when we were talking about the different types of oxidation for the fatty acids, alpha oxidation is only for the branch chain fatty acids. Therefore, the problem here is going to be impaired ability to degrade branch chain fatty acids. That's going to be the main issue here. The rest of the function of the peroxisome is generally fine in this disease. So the labs here are going to be just limited to elevated levels of phytanic acid, which is a type of branch chain fatty acid. Again, that makes sense. You can't degrade it, so serum levels are going to go up. Symptoms are ataxia, which is balance problems, ichthyosis, which is scaly skin, cataracts, which can cause night blindness, and shortened toes, specifically the fourth toe. The treatment here is very interesting because this is one that actually has a treatment that works rather well. That's dietary. And the idea here is to just avoid branch chain fatty acids. So since this is the only thing that you can't degrade, you just avoid them in the diet so that they don't accumulate inside the body. But if they do and it becomes severe, then you can do plasmapheresis to remove it. All right, and our third and final disease is X-linked adrenoleukodystrophy. So obviously this one is different than the other two. It is X-linked recessive, not autosomal. And here the defect is a mutation in this ABCD1 gene. So this ABCD1 gene encodes for the ABC transporter, and this transporter is important specifically because it's on the peroxisome and it's important for uptaking very long chain fatty acids from the cytoplasm into the peroxisome so that the peroxisome can chop them up, break them down, and degrade them. So therefore, if there's a mutation here, that means you're missing this transporter. That means you can't uptake very long chain fatty acids from the cytoplasm and break them down. That means that the very long chain fatty acids are going to build up inside of the cell. And this specifically seems to affect specific tissues. So adrenal glands, that's why it's adrenoleukodystrophy. The white matter of the brain, so that's where the leuko of the name comes in, and the testes, that's not part of the name. So adrenoleukodystrophy, dystrophy, the adrenal stands for the adrenal glands, the leuko stands for the white matter of the brain. So the labs here, again, are going to be specific to high levels of very long chain fatty acids in the serum because that's the molecules that are not getting broken down. And the symptoms are going to be pretty significant here because of the effects on the brain and the white matter. So this is going to cause progressive neurological deterioration. And of course, the effects on the adrenal glands is going to cause adrenal insufficiency. And again, this is one of those where the prognosis is very poor, treatment is supportive only, and this usually progresses to death. All right, so let's wrap up this video, taking it back to where we started. Remember our zome structures, the lysosome, peroxisome, and proteasome. For those of you who like summary tables, here's one that compares these three structures with their key features, their functions, and their clinical correlates. So of course, in this video, we talked about the peroxisome, so you can feel free to review this now or use it in the future for reference. Okay, that's it and that's all. If you want to learn more about this topic, you can check out other videos that I've linked here. And if you guys enjoyed this video, please don't forget to like, comment, and share it with others. I also want to invite you to subscribe to this channel so you don't miss out on any upcoming content. But for now, thank you for watching, and I'll see you next time.